appreciate the invitation. Can you hear me?
No, it's not working.
Okay, appreciate the invitation to come visit MSU and to have a little bit of a Midwest experience. I don't get that very often. I have family on the East Coast and I live on the West Coast, and I just fly over you guys most of the time. But it's very beautiful this time of year, and I'm enjoying my visit. So I want to start with a story. As Mark said, literally, this, not me, I wasn't written up, but my sheep have been written up in various places, written about in various places. And back 5 years ago or so, the research got a lot of publicity. You may have heard of it for various reasons, not all of them good. But some of what came out of it was a short story that was written in the New Yorker by, it was called Sheepish, and it was written by Paul Rudnick. And it goes, "Enough already. I'm Troy, a gay sheep, and I'll tell you the truth. Although I'm conflicted about calling myself a gay sheep because I don't like to think that my sexuality defines me. Let's just say I'm a sheep who happens to be gay. Being gay is just a simple biological fact, like having a fleecy undercoat, or bleeding while you're being shorn, or getting aroused whenever you see a bulky turtleneck sweater." It goes on in this vein, but the point is, I think Mark is presenting you with a series of lectures to give you the science behind the biological influences on sexual orientation. And my goal today, and hopefully from those lectures you'll make up your own mind based on those facts, how much of a role biology plays. But my goal today is to tell you about my sheep research and how we believe, at least in sheep, biology has a big role to play. And that some rams are attracted to other rams, and we think it's in part because they're born that way. And so I want to present that evidence to you today. What we measure in sheep is called sexual partner preference. It's the preference for one sex or the other, or even both. You know the terminology: homosexual, heterosexual, bisexual. It's not quite the same thing as sexual orientation in humans, or at least I don't believe it is. It's a pattern of sexual attraction, what we're looking at in sheep. But where it differs is it doesn't have the human qualities of emotion, romantic love, or even a sense of identity. As far as we know, sheep don't self-identify as homosexual or heterosexual. The thing about sexual partner preference, and that's really obvious and to keep in mind, is that it differs between the sexes. Typically, males are attracted to females, and females are attracted to males. So we exploit this sex difference to look at it in various ways. Well, why study rams? Mark's already kind of alluded to this, but there's various reasons for why this seems to be a good model of sexual orientation. One is that rams exhibit natural variations in their preferences. They range from exclusively heterosexual to exclusively homosexual. The rodent models, which he's skeptical about, really look at a kind of bisexual behavior, which comes about from early hormone manipulations. This is a little bit different. It's naturally occurring. The behaviors, as for all animals, are verifiable. We can watch these animals. We can record their behaviors, which we do. We can score their behaviors. We don't have to rely on them telling us what they're doing. Again, there's no self-identification here. The preference of a ram stays with the ram for its entire life, as far as we can tell. So these are long-lasting. They're enduring. They're not just a one-time affair or a situational kind of behavior because they've all been crowded together or something. We think it's truly something that characterizes them. And lastly, one of the advantages is that it's a great model because we already know a lot about the reproductive physiology of sheep. We know a lot about their behavior, and we know a lot about their development, so we can build on all of that. Well, let me tell you the story of how all this began. Our study started as a collaboration with agricultural scientists at the U.S. Sheep Experiment Station in Du Bois, Idaho. It's in a godforsaken, isolated part of Idaho. It's pretty. It's kind of high deserty, but it's pretty. That sheep station had an interest in sexual behavior of rams. Why? Why would you be interested in sexual behavior? Well, farmers are, because most farmers only use one ram to breed 50 or 100 ewes. So if that ram is not performing, as they say, that's an economic burden to the farmer. It's not a good thing. So the sheep station would routinely test 500 to 600 rams a year and score them on the basis of their libido to identify high libido rams that they could provide in auction to the farmers. And they also were doing studies of breeding those rams, that kind of thing. That's a lot of rams to test in a year. And we were fortunate enough to adapt some of their methodology in order to identify rams for our studies. So it actually takes 2 tests and over 2 years to identify a ram's sexual partner preference. The first test is the test that they were routinely given. It's called a performance test or a serving capacity test, how well these rams service ewes. That's what they call it. Okay? And it's a 30-minute test in which the ram is exposed to 2 sexually receptive females. And for that 30 minutes, all of their behaviors are recorded, their courtship behaviors, mounting, ejaculations. And the test is repeated 18 times for a total of 9 hours over a 1-month period of time. And you can see these people sitting there recording several rams and their behaviors. At the end of that first test, all we're going to know is some of our rams are going to be sexually active, and some of our rams are going to be sexually inactive. All right? About a quarter of them are going to be sexually inactive, actually. But to determine what their preference is, we've got to take a subset of each of those 2 groups of rams and give them a second test, a sexual partner preference test. And this test is a choice test. In this test, it's also a 30-minute test. The ram is exposed to 2 sexually receptive ewes and 2 rams. And then their behaviors with both of them, we watch whether they mount females or mount the males. We record how many times they do it. And that test is repeated at least twice in the first year, sometime a couple months after the performance test. And then we let them go ahead and manage their rams for a whole other year, bring them back in and test them a year later to make sure that the behavior is stable. It's going to occur again. At the end of the second year, what happened is the rams were then trucked over to Oregon. So this is a very involved process where we studied them further, and we often gave them additional preference tests. So at the end of all this testing, we end up with 4 categories of rams. We end up with rams that are exclusively female-oriented or heterosexual. We use this terminology, female-oriented, male-oriented. But really, it's a heterosexual ram. These are rams that in those 2 tests, those 2 series of tests, only and ever mounted females. And this is based mostly on their mounting behavior. The second category are exclusively male-oriented rams. So this means that these rams in the first performance test didn't do anything with females, and in the second preference test only mounted and interacted with males. So they're exclusively male-oriented. And then there's also, as you might expect, some bisexual rams that might have mounted females in the first, males and females in the second, various combinations. They're the bisexual rams. And then there's still a proportion of the rams that really showed no sexual behaviors, the asexual rams. We were only interested in the top two. These other categories are very interesting, but we thought we could study these two exclusive categories and try to make some comparisons between them. This shows you the proportion of rams in each of these categories from a group of almost 600 sheep that we tested back in 2001. As you might expect, the greatest majority of them are female-oriented or heterosexual, a little over 60%. But there's a fair amount of bisexual behavior, bisexual animals, and asexual animals. The male-oriented proportion turns out to be about 8 to 10% of the population. I've listed 3 studies down here to make a point that these are 3 independent studies. They were conducted with independent populations of sheep in different areas of the country. Ed Price in California, Ann was in Idaho, and we were in Oregon for this. And in all 3 of these studies, reported about the same proportion of animals in each of these categories. There can be some shifts from year to year, but it's fairly about the same. Now, I want to make one more point about these rams and their sexual behavior. They show an atypical sexual attraction for a male, and almost what we would call a female typical attraction. They're attracted to males. But the behaviors that they perform are all very male typical. They show all the sequence of courtship and mounting behaviors that any heterosexual ram would show. And it's really a pretty stereotyped set of behaviors. They nudge and they nibble and they nudge the other animal. They do something called a foreleg kick, where they kind of kick at the animal they're going to mount. Then they mount them, they thrust, and they occasionally even ejaculate, but they do it with another ram. So let me, if it works, show you a tiny little movie of this where you'll see some of these elements. So there he is, nibbling and nudging. Oh, in the foreground is a ewe. The lighter one is a ewe. In the background is another ram, and that's the test subject ram. He's nudging around. I'm getting a glare. I don't know if you can see, but if you look at his left foreleg, he starts to do a little bit of a knee kick. There's a foreleg kick. Foreleg kick. He's not interested in the female at all. He's been in there for 30 minutes. He mounts, thrusts, he has an erection, and that's the typical kind of behaviors, only he's showing it to a male. So a reasonable question to ask is, could hormones be involved in this? Could sex hormones be involved in this? Sex hormones control a lot of other sexual behaviors. Why not sexual partner preferences? To explore that question, let me first tell you about how the kinds of effects sex hormones have. They're thought to have, we know that they have 2 distinct mechanisms through which they work. The first mechanism is called an organizational effect. And these are effects that sex hormones have early in the life of an animal. And they're the effects that essentially wire the brain, hardwire the brain. Some of these effects, I see Cheryl in the background, can occur even a little later during puberty as well. But these are thought to sort of set up the circuitry and the wires in the brain. Then there's a second mechanism through which sex hormones act, where they act on the fully developed adult brain, called activational effects. These occur, like I just said, in adulthood, and they kind of flip the switch. They activate the circuits that either turn on or turn off behaviors. So these are the 2 mechanisms by which sex hormones act. And most of the evidence favors the idea that it's the organizational effects that influence sexual partner preferences, at least in the studies that have been done in rodents. So how does this work? Well, neuroendocrinologists have known for quite a while that sex hormones program behaviors. And the way this seems to work is that the brain really starts out as a bit undeveloped. And if the developing animal is a female, the brain will develop along a default pathway without any nudging from hormones of any kind. And when the animal reaches adulthood, she'll show female sexual behaviors and sexual partner preferences under the influence of her ovarian hormones. Now, if the developing animal is a male, early on the testes come into play and secrete testosterone, and this changes the development of the brain. And now, when the animal is an adult, he'll show a set of male typical behaviors, sexual partner preferences. There's three other important aspects of this process that I should mention. One is that the effects take place during a critical period. And during this critical period, the brain is susceptible to the actions of sex hormones. The critical period for rodents is usually the first week of life, first few days to the first week of life. That's when you can change the testosterone acts to change that development. In sheep and in humans, the critical period is actually during the first or the second trimester of gestation. And so it takes place during fetal life. The second aspect of this that should be mentioned is that in many species, testosterone has to be converted by the enzyme aromatase to estradiol. So remember that name, aromatase. It's an enzyme. It's going to come up a little bit later. Mark mentioned it before as well. It converts a male hormone, testosterone, to a female hormone, estradiol. But the brain really doesn't care about that kind of a distinction between these two molecules. They're just active molecules to the brain. They have independent effects, somewhat complementary, and they both have effects in terms of carrying out the male program, or the masculinizing program, as you would say. And the third thing is that these effects of sex hormones are permanent. Once you've committed to a pathway, there's no turning back. The only way that you can change the development is if you manipulate the sex hormones early in the critical period. And you can do that. So, especially in a rat during the first week of life, if you castrate the males or block the aromatase activity or enzyme in their brain, you can convert their what would have normally been male behaviors into female behaviors. And in the females, during their first week in a rat, if you treat them with testosterone or estradiol, you can convert their ability to show from showing female behaviors to showing male behaviors. So this is called the organizational hypothesis. And it gives you a background. Well, in 1984 or so, Roger Gorsky showed physical evidence that that sex hormones actually change the structure of the brain. And what Roger did was he looked at he first looked at adult rat brains. And for those of you that aren't neuroanatomists, what he did was slice those rat brains into very thin sections, stain them with a dye called thionin, and then that dye marked cells in the brain, and he could look at the arrangement of the cells in different brain sections. And he could compare them between males and females. So this is a section of a male and a female brain. And he looked specifically in an area called the preoptic area, which is a part of the hypothalamus which is known to be involved in male sexual behavior. And what he found was that there was a cluster of cells which was larger in males than in females. And I circled it, and you have been staring at it for a while. So that cluster is larger in males than in females, and he called it the sexually dimorphic nucleus of the preoptic area, or the SDN. The way he showed that sex hormones were involved in regulating the size of that SDN is that he treated females early in life, in the first week of life, with either testosterone or estradiol, the same hormones that would have changed their behaviors. And then he looked at their brains, and you can see that he looked later when they were adults at their brains, and you can see that their SDN is large, just like the SDN in the male. So it masculinized their brains. So this was evidence that sex hormones were changing the structure of the brain. Well, one last piece of sort of background information, about 10 years later, not quite, in 1991, Simon Levay looked at autopsy brains from straight and gay men and found that there was a structural difference in the brains of gay and straight men, very reminiscent of this SDN. He looked in a similar area. It's called INA3, and that stands for the third interstitial nucleus of the anterior hypothalamus. Same sort of area that's involved in sexual behavior. And this shows you the brain sections, the autopsy sections that he stained with a dye, and INA3 was larger in these representative sections, at least in area, between a heterosexual man and a homosexual man. Then he calculated the volume. And I want to tell you about how you calculate volume because with sheep we do this a lot. So these are three-dimensional structures, these collections of neurons. They extend back, forward and back in the brain. And to calculate a volume, you actually have to slice through the entire structure, or at least sample through the entire structure. And then you take the average volume of the average area of that, which you can just measure. Whoops. The average area, which you can measure here, and you multiply it by the total length of the collection of neurons. And when you do that, you can calculate the volume. That's what he did, and he found that the volume of INA3 was twice as large in a straight man than in either a gay man or a woman. Now, you got to understand the populations he used. They were autopsy brains from presumed heterosexual and presumed heterosexual men and women, and they were autopsy brains from gay men that died of AIDS. So this, although this was exciting data, it was very controversial because we didn't really know that these categories were truly the categories that they were thought to be. We didn't know if AIDS affected the size of the area in gay men. And there's been some studies since then to suggest that AIDS has no effect. Simon Levay showed that himself. And then a second study was done by Bill Bine, which largely replicated this effect, or this difference. So these data suggested that INA3 volume not only correlates with the sex of the individual, but it correlates with their sexual orientation. And with this study, it became possible to think that maybe brain structure could influence sexual orientation. Because of the reasons I just told you, that there was skepticism associated with it, it hasn't been highly replicated, there was a need to somehow replicate it or show it in other species. Well, I've told you that we can identify rams that are heterosexual and homosexual, and I've told you now that the brains of straight and gay men are different, so obviously it was of real interest for us to look and see whether our ram brains were different. So we formulated, or formalized this, I guess, as a hypothesis. And our hypothesis was that the brains of male-oriented rams undergo less masculinization than the brains of female-oriented rams. It's specific about what we thought was going on, not mechanistic though. And from that, we predicted that the SDN in male-oriented rams is going to be significantly smaller than in female-oriented rams and similar to use. Very reminiscent of what I just showed you for the human study. Well, we didn't know whether there was an SDN in rams. Nobody, or in sheep, no one had ever reported one. What we did know is that the SDN should be located around, or in and around the preoptic area. That's where it is in almost every other species. That's where it is in every other species. And we knew from studies in other species that this enzyme aromatase, remember, the enzyme that converts testosterone to estradiol, we knew that that was highly expressed in that area. So we felt we had two tools to go looking. One, we could slice through the sheep brain and stain it with a dye to look for the pattern of cells. And we could also use a probe. We used a radioactive probe, slightly radioactive probe, to look and see where the aromatase gene was expressed. So these are our two tools. Slice through those ram brains. And when we did that, we found that there was a cluster of cells sitting right in the middle of the preoptic area. And this smudge here is the dye-stained brain section, a picture of the dye-stained brain section, and that's the collection of cells that's there. We've analyzed it in many other sections. And this shows you where that aromatase probe told us the aromatase gene was localized. And you can see they overlap considerably. So these are our two identifiers for the nucleus. Now this doesn't tell us, I say it's an SDN. It's very suggestive that, well, we have something in the right place. So the next thing we had to do was measure the volume. Remember, we had to cut through the entire length of that collection of cells and multiply its average area by its length. And when we did that, we found that the volume of our female-oriented rams was 2 to 3 times larger than the volume in either the male-oriented ram or the ewe. And though I'm not showing you the data, we also measured the aromatase signal, as it were, and that was also higher in female-oriented rams than male-oriented rams or ewes. And these two guys, ewes and male-oriented rams, were fairly similar. So again, this was very reminiscent of what Levay showed and Bill Bine after him in the human brain, and served to kind of, in another species, lend some support for that. Well, what does it really all mean? What it means to us is that we show that there's an association between natural variations in preference, a brain structure, the SDN, and the expression of a gene, in this case, aromatase. It doesn't say how they act together, but that there's an association. And that rams that prefer other rams show a greater size SDN and more aromatase than rams or ewes that prefer other rams. I might have said that wrong. Rams that prefer females have a greater or larger SDN and more aromatase than rams that prefer other rams do. Well, how's this association arise? That's the next kind of question we have. And there's two possibilities. The male-oriented rams could maybe have been exposed to lower levels of testosterone during development. The lower testosterone that they were exposed to maybe caused a smaller SDN to develop, and that influenced their preference for other rams. But it's also possible that the behavior could have developed first because of other reasons, either experience or learning, and then continued performance of that behavior may have changed the size of the SDN. So which is it? Does the SDN develop first or the behavior? It's a chicken-egg kind of question. The trick here is that we can't answer this directly because we can't know whether a ram lamb is going to grow up to be a male or a female-oriented ram. If we could study the development of their brain over time and get some sense of that. So remember, I told you that a lot of this stuff is different between males and females. So what we can study is that we can study and ask questions about what controls the sex differences in the development of the sheep brain and make inferences from that back to how we think things go for male and female-oriented rams. So one of the first studies we did was, and questions we asked, is whether the SDN volume is affected by adult hormone levels. We wanted to make absolutely sure that these different groups didn't have different levels of hormones circulating at the time that they were sampled, and that was the reason that their brains were different. So to do this test, we gathered together, did our testing, and gathered a group of male and female-oriented rams and a separate group of control ewes. We gonadectomized all the sheep, and then about a month later, we started treating them with the same doses of testosterone, so they would all have the same blood levels of testosterone. And at the end of 3 weeks, we euthanized them and we measured the SDN volumes. And what we found that these animals, remember, all have the same or equivalent levels of testosterone in their blood. And even when you equalize the testosterone, male-oriented rams still had an SDN that was significantly larger, almost twice as large as that in male-oriented rams or ewes. So this tells us that it's not due, the size difference isn't due to the levels of hormone in their blood. So the next question we asked had to do with, well, maybe we could show whether or not the SDN develops before the animal's born. If it develops before the animal's born, before it's interacted with any other animals, before it's shown any behaviors, then that would be evidence that it's the first thing that develops, and then the behavior is somehow influenced later. So this shows you a developmental timeline, and it plots the levels of testosterone in the blood of male and female sheep fetuses across gestation in days. And for a sheep, their gestation is about 150 days long. Gonads develop about day 30. And up to this time, we believed that the critical period, remember the period that when the brain is most susceptible to the effects of sex hormones, constituted a period from day 30 of gestation to about day 90 of gestation. So if we reasoned that if we wanted to go looking for an SDN before birth, that if we look sometime after that critical period, after testosterone seems to drop in males and rise a little bit in females, and so out here it's about equivalent in both sexes. So if we look out here, maybe we'd see an SDN. And that's what happened. Our hunch proved correct. When we looked out at gestational day 135, we saw that there was an SDN, and it was larger in males than in females. And that's illustrated in these brain sections. And this shows you the thionin stain section. It's more obvious in the sections that use the aromatase probe. But of course, we had to measure the volume as well. And this shows you the average volumes for males and females. So it's larger in males than in females. For comparison, we also looked at the size of another nearby nucleus called the AVPV. And we found that in sheep, that nucleus is not different between males and females. And so that indicated to us that the sex difference is specific for the SDN, and wasn't just due to the way we were handling our tissue or some artifact of our methodology. Well, the next thing we wanted to know is if this sex difference exists, does it exist because the animals are exposed to testosterone? So we asked, can prenatal testosterone exposure masculinize the SDN? in females. I guess this shows our experimental setup, a developmental timeline. You can't directly treat the fetus in a long gestation animal. What you have to do is give injections to the mother to expose the fetus. So we gave the mother weekly injections of testosterone so that the fetus was exposed to the hormone from days 30 to 90. We call this the long T treatment. Control mothers just received oil injections. Then on gestational day 135, we surgically delivered the fetuses, and we looked at their gonads to determine what their sex was, and we looked at their SDNs and measured their SDNs and their genitalia. And what we found was that we could replicate the sex difference we saw before. Males had a larger SDN on average than females. The prenatal exposure to testosterone didn't affect the size of the SDN in males, but it dramatically enlarged the SDN in females. So this shows that testosterone exposure during that critical period masculinized the female SDN. So it tells us the egg comes first. Differences in SDN volume develop first, and then they may later influence the sexual partner preferences. I want to talk about the back side of the sheep for a second, because it comes up in a point I want to make in a second. So in addition to masculinizing the brain, the SDN, testosterone exposure during fetal life also masculinizes the external genitalia. So a normal male, or control males, have a penis and a scrotum, and females have a vulva and ovaries. But those females that were exposed to testosterone during the critical period, their genitalia develops into male genitalia. They'll show a penis, and they'll have a scrotum, although their scrotum will be empty because they still have ovaries. And males exposed to testosterone during that long T treatment won't be any different. Normally, the sex of the brain and the sex of the gonads match. And that's true here, too. These females had a male SDN, and they have male genitals. But that's not true for our male-oriented rams. Remember I told you, they have male genitalia. Functioning male genitalia, they produce sperm, that side of them is typically male. But they have a female typical SDN, so their genitals and their brain don't match. So how can that occur? Well, one way that might occur is if there's a different critical period for the differentiation of the brain than there is for the differentiation of the genitalia. And we went looking for that by thinking, well, let's split our treatment period in half. And that's shown in this experimental setup. What we did was we used our testosterone injections, and in one group of pregnant animals, we gave it during the first half of that long critical period. And we called that early T, from day 30 to 60. And we gave a second group injections from day 60 to 90, and we called that the late T group. And then control animals, again, received oil. And the fetuses were delivered, again, on day 135 of gestation. Now, this is a complicated experiment. Males and females, different treatments. So I want to talk about the females first. And this shows you what happened with the genitalia. So this is normal control females. Turns out that the early T treatment masculinizes the genitalia in females, whereas late T treatment had no effect. So this tells us that the critical period for the genitals is early. It occurs from day 30 to 60. When we looked at the SDN of these same animals, we found that the early T treatment had little effect, and the late T treatment had the masculinizing effect. And you can see that in these representative sections here of control, early T and late T. And you can see it also in our more thorough volume measurements, where really the late T had the dramatic effect to enlarge the SDN. And the horizontal line that I'm showing you there is the average for a control male. So this says that the critical period for the SDN occurs later. Another important thing that's demonstrated by this is that there can be a mismatch between the genitals and the brain depending on when they see testosterone. And that's illustrated here. So here's the control situation, they match. Here's early T, you have masculinized genitalia, but a feminine SDN, so that's a mismatch. And here's late T, you have a masculinized SDN, but a feminine genitalia, that's also a mismatch. So it's possible to mismatch, and there's probably multiple critical periods. This is the critical period for the genitalia and the brain, and the SDN. Well, what about the males? The males, you would expect, giving them testosterone, they already make testosterone. That testosterone shouldn't have much of an effect. And that's true for the genitalia. Neither the early or the late testosterone seem to do much. But when we looked at the SDN, we saw a very unexpected effect. Early testosterone actually feminized the SDN, whereas late testosterone had no effect. And you can see that here, again, in the brain sections that illustrated, the dye-stained brain sections, and here in the volume measurements that we did. And again, I put a horizontal line here to show the average of the control females. If you apply this idea that inappropriately early testosterone exposure can feminize the male brain, we could apply that to the male-oriented rams, and maybe it can explain what we see in male-oriented rams. Because remember, male-oriented rams have a lot of male-typical characteristics. Their genitalia are male-typical. Their actual sexual behaviors are male-typical. I didn't tell you about it, but a lot of hormone responses are male-typical. They won't show female behaviors, so they're not really somehow feminized in their female sexual behaviors, but they show a female preference. So maybe this can explain one way in which male-oriented animals come about. And so our hypothesis, and you probably can't see this all that well, is that, and what we've shown now in fetuses, using the fetuses, is that early T excess acts to hypomasculinize or feminize the SDN. And we would predict that that causes animals to have or develop same-sex preferences, partner preferences. That's what it says in the black. I can't see that. And this is what we'd like to test. But this is going to require us to take these animals that were treated early in gestation and then let them grow to adulthood, which is almost an 18-month period of time, and then test their sexual preferences. So, what do we know? What do we know about rams? Why are some rams attracted to other rams? Well, we don't really know the answer to that yet. But most of the evidence that we've collected, and all of the evidence I've presented to you today, suggests and strongly points to the idea that the prenatal hormonal environment plays a strong role in influencing those sexual partner preferences. There may be natural variations among individuals, or there may be environmental factors like stress or nutritional factors, undernutrition, which could affect the levels of testosterone that the fetus is exposed to and cause something like an early excess exposure to testosterone. And so where things stand right now is that this prenatal hormone environment plays a very important role. I want to make one final point, and that is that what this research is all about is trying to understand the connection between brain physiology and sexual partner preferences. It's not about trying to change any of this, and it's not about a direct correlation with human behavior either. Ultimately, what these studies tell us about is the sheep. But we really think that animal experimentation is important, because what it can help us do is formulate questions and hypotheses that we can then test in other species, and maybe even help us frame questions that we can ask about human sexual development and development of sexual orientation. So none of this research would be possible without the hard work and input of a lot of people. And I especially want to acknowledge my laboratory, members of it past and present, who have been part of these studies, and to acknowledge the invaluable assistance of my colleague and collaborator at Oregon State, Fred Stormshack. You guys have heard of Oregon State, haven't you? I think they're now ranked 10 in the coaches poll or something like that. I don't know. Anyway, these guys are great. All of our sheep are down in Corvallis. I'm up in Portland at a medical school on top of a hill. We don't have any sheep pastures. So these are the guys that do a lot of the handling of the sheep. And then I've had the good fortune and privilege to work with a number of other people, the people at the U.S. Sheep Experiment Station out in Idaho. The early work was really dependent on their cooperation. And we've had collaborations with our friends at University of Wyoming and even at UCLA. Someone you saw earlier, Eric Filane, was involved in some studies. And then last but obviously not least, I've got to acknowledge my funding agency, which was the National Center for Research Resources, which is part of the NIH, which has now been subsumed into the office of the director of NIH. And so it's got this really nasty acronym associated with it. Finally, I'd like to thank you for your attention and to take any questions.
So thank you. So I'm sure there are going to be lots of questions, and so please raise your hand. I'll try to get to you, and try to use the microphone so everyone can hear the question you have.
So I know that twinning occurs pretty often in sheep, and I was wondering if you examined any partner preferences in fraternal twins.
It's a great question, but no, we haven't been able to do those kinds of studies. We haven't really studied the behavior in that many animals to be able to make those correlations.
Let's hear them.
Has there been any speculation about, I know there's been, your research has been figuring out the correlation between the SDN and hormonal influences in gestation, but do you know if there's any research done or speculation about whether there's an actual causal effect between the SDN's structure and size and sexual orientation, or whether it's just correlation?
At this point, it's just correlation, and that's really the big question: is there a causal link? And some of the studies that we've initiated with our collaborators at Wyoming, their purpose was to destroy SDN and see whether in a heterosexual animal, and see whether it affected their preferences. Those are very difficult studies. People do lesion studies in rats all the time, but the skull of a ram is about this thick. And even people do a lot of stereotaxic surgery in ewes, and that my collaborator who is doing this didn't realize the big difference, and so it turned out to be a very difficult problem. And so there's two ways that we try to approach it. One is that if we could show that you can somehow affect preferences by altering the nucleus itself. The other way is to give these prenatal treatments and show that we affect their behavior later in life. And we haven't been successful at that yet. This is one of the studies we'll try to do.
As you do this experiment on the males who are exposed to testosterone to reduce their SDNs, are you going to also look at the females who got exposed to testosterone and had their genitalia and SDNs masculinized? And related question, what is female sexual behavior in a sheep?
Well, we probably won't look at the females. We've done some of these studies before, and the groups just get maintaining all the animals are a lot of effort. So we'll probably just look at the males. But your question about what is the behavior like in females? Female sexual behavior in sheep is really nondescript. I don't know if you know about sexual behavior in rodents, for instance. They have a whole set of soliciting behaviors. They ear wiggle, and then they lordose, and they run around and they lordose for the male. Sheep kind of just let males mount them. They'll at least get close to them. They will get in the general area of the male, but they don't show a lot of demonstrative behaviors other than they don't reject the male. I mean, there are some things they turn, but they may wiggle their tail, but it's not very demonstrative. And as far as, I don't know if you were asking this as well, but females don't show a lot of what we call heterotypical behaviors. They don't show a lot of male behaviors. Like you don't see a lot of female-female mounting in sheep. Some species you see that. Cattle, you see a fair amount of female-female behavior. You see it occasionally in female sheep. So no one's ever looked around to see whether there are female-oriented females, as it were.
Chuck, I'm just wondering what your thoughts are on how and why early high levels of T would feminize or hypomasculinize, as you put it. Are we just talking about an inverted U-shaped function for testosterone actions on the brain, or something more exciting?
Well, we're starting to look. It could be affecting the HPG axis. So we have some data that LH and testosterone in those animals later, so it's exposed early, but really the time when androgens affect the SDN occurs a little bit later. So you could be setting up a situation that you somehow retard the HPG axis development, and now they're seeing less testosterone during that 60 to 90 day period. We have some hints of that, but I'm not convinced that that's the real thing that's going on. We also have some evidence that early T is suppressing androgen receptors. And so there could be something at that level, or there could be something at the level of genes that are affecting survival, for instance, or something like that. And that's kind of, there's actually at least 3 levels. It could be happening at the hypothalamic level, at the gonadal level, something could be going on, or it could be happening directly on the brain to alter the developmental profile. So there are the 3 things we're going after, trying to eliminate one or the other. Maybe they're all involved, I don't know.
And relating to that, and to relate back to Dennis McFadden's talk, he too is wondering whether there are different critical periods for testosterone to affect different things, because how is it that testosterone seems to be affecting otoacoustic emissions and not genitalia? Well, his suggestion is that It could be that the otoacoustic emissions are eliciting testosterone later. And what I find intriguing, if you were talking about how the animal models can suggest other hypotheses, the other thing he pointed out is that gay men, on average, have larger genitalia than straight men. And so your hypothesis that higher early testosterone, I mean, I know you didn't see an effect of early testosterone on your males, but on the other hand, when it masculinizes female genitalia, it's much more effective to give it early than late. And so, I mean...
Yeah, that's absolutely right. But, and I got to admit that we didn't measure penis length. We just looked to see that there was a scrotum and a penis, and that it wasn't somehow dramatically altered in the male. Now, it could be...
Just as the difference between gay and straight men isn't dramatic, it's not.
Yeah,
it's an average difference.
If you looked close enough, you might see some physical evidence that early T had some other effect as well.
Right.
But you're right. That hasn't escaped me, that idea that there's some data that suggests that gay men have been exposed to more testosterone, not less.
Right, and like your gay rams, they show lots of male behaviors, right? So, Eric, do you still have a question? Yeah. We'll go from the base to the sublime here. Eric's going to ask about the brain, I bet.
Actually, I was going to ask about correlation versus causation. So if you manipulate STN with early T and then watch for behavior later, my first thought would be that the size of the STN is just another dependent variable, and that you've got some third variable that you're manipulating with the early T.
It's a good point. I mean, that could be true as well. I mean, it doesn't prove that it's causal. I think the only truly, it's dancing around, but the only truly causal thing would be to manipulate the SDN directly and somehow change the behavior. And like I said, we're trying to do that, but that's very, very difficult.
Other questions? I'll ask one more. And when Paul Macy was here, he was talking about this idea of a gene that when it occurs in a female increases their reproductive output because if it makes them especially attracted to males. I know you say that ewes don't seem to be playing a very active role, but his idea was that a gene that might increase reproduction in females, if that same gene occurred in a male, might make them more likely to be attracted to males. So do you have any idea whether the sisters of your male-oriented and female-oriented rams, whether they differ in their fecundity?
Is it the sisters or would it be the mothers?
Well, I
Or
mean,
maybe both.
you could do both, or sometimes even, depending on how good the breeding records are, the maternal aunts of the different kinds of rams.
So the breeding records that I've looked at suggest that the mothers are not any more fecund than other mothers, but I can't tell you that for sure I have the complete breeding records. Well, it's not that I don't have the complete breeding records, it's when they decide to get rid of the mothers. So I have breeding records up until the time that I get the rams, and I don't think that's the best record.
So if some of the dams, maybe if they weren't commercial animals that were being allowed to live the lifespan, maybe they would reproduce for longer.
Right. And the other thing is through all this that we've, for instance, the idea that it's passed on through maternal lines, we've never had two males that were the same orientation that came from the same mother. It just never worked out that way, or that came from related mothers or whatever. So, and we weren't setting up the breeding situation. If we could have set up the breeding situation, you could have tested some of that.
And so you think it's conceivable that this is like entirely an environmental or epigenetic influence, and maybe it might not be a genetic? There might not be a genetic?
No, I don't think I could close the book on anything.
Okay. Good. How about one more question? So I can get my exercise in.
So is there a correlation between, depending on the length of the life of the ewe, the more males she has, the more likely the later born males are going to be more attracted to
Yeah.
other males?
We've done those calculations too. I don't know if Mark remembers, there was a point where I thought there was, but as I collected more data, they're almost, there's definitely no correlation between more older brothers and their preferences. So we didn't see that. And most of our sheep are going to be twinned or even triplets. So that sort of confounds it as well. But yeah, no, there was no evidence of that.
Okay, all right, well, all right, one last question. Okay. One last question.
So you mentioned earlier that ewes really don't really show a lot of sort of mating behaviors. Question, was there any study at all about sort of male rams that would prefer to be mounted?
I
mean, is
I expected that somebody would ask this question.
Yeah.
So, no,
And it's
So
probably
it's
a
not
bad species
just-
to study for that.
pardon?
It's probably a bad species to study for that because, as you said, ewes the females don't really show a lot of.
But you're asking about the males. Are
Yeah.
there males?
Yeah.
What do we know about the males that get mounted? So when these males that we test them with they're just males. They're picked out of the flock. They're put in there. They're not particularly the males that get mounted. But when animals are in an enclosure, in a pen, or in a field, there are certain males that do get mounted. And nothing's been done to study those animals. That's a phenomenon that happens for cattle too. Apparently it's pretty common in cattle, and they call them buller cattle because the bull males mount them a lot. And they think that they're actually, in terms of cattle, are somewhat feminized males. But again, that's not been rigorously or scientifically tested. And in sheep, certainly nobody's looked at that. But I can tell you that none of the animals that we have studied as male-oriented were also males that got mounted. So they don't go both ways, I guess.
Okay, so there are more questions left to be answered, apparently?
Oh, lots of
And so you want to come back 2 weeks from tonight and hear more? All right, thank you.